Hi, and welcome to the digestive system. I hope everyone is doing well. Um, Today, we're going to talk about listing and describing the functional anatomy of the organs and the accessory organs of the digestive system. We're going to discuss the processes and the control of the ingestion, propulsion, mechanical digestion, chemical digestion, absorption, and defecation. We're going to talk about the roles of the liver, the pancreas, and the gallbladder within digestion, and we're going to compare and contrast the digestion of our three macromolecules. So the digestive system is divided into two categories. We have the alimentary canal. The alimentary canal is actually the canal that the food is traveling through. Okay, and its job is to nourish the body. This is a one-way tube, and it's about 25 feet long. And then there's some accessory digestive organs. And these organs, the food's not going to travel through them. However, they actually are going to secrete something or store something that helps with digestion. Okay. So, for instance, in the alimentary canal, you have to think about, the food is going to enter your mouth. We're going to swallow it, and it's going to go down the esophagus. Okay. It's going to go into the stomach. Then it's going to travel into the small intestines. Okay, so the first part of the small intestine is called the duodenum. Then we have the jejunum, and then the ileum. And then once we come out of the ileum, we're going to end up in the large intestine, also known as the colon. We're going to travel up the ascending colon, across the hepatic flexure, which is right here, then through the transverse colon to the splenic flexure, down the descending colon, the sigmoid colon, and then out the rectum. Okay. So our accessory digestive organs would be like here in the mouth. We have all of these salivary glands, like the parotid glands, the sublingual glands, and the submandibular glands. They're going to secrete saliva to help with digestion. Our liver here is going to secrete bile. That bile then gets stored in the gallbladder for later use. And then we have our pancreas, which our pancreas is this yellow part, and it's sort of right tucked behind that large intestine. And it's going to break down the fats, the proteins, and the carbohydrates. So our GI tract, if we start sort of on the inside, the lumen is the hole. Okay. And then out from there, we're going to find the epithelium, the lamina propria, and then the muscularis mucosa. All of that makes up the mucosa layer. Okay. Then we're going to have all these glands hanging out. Okay. We're going to get to the muscularis. And the muscularis is pretty cool because we have circular muscle and longitudinal muscle. And this allows the GI tract to be able to do what's called peristalsis. Peristalsis is sort of this milking motion to help move the food down through the alimentary canal. Then we have on the outer part here, we have the serosa. Okay, so we have the epithelium and then the areolar connective tissue. Within there, we have the mesenteric plexus. The mesenteric plexus is going to be our nerves. Okay? And then we're going to hit the mesentery. And the mesentery is going to be that covering to sort of cover over that GI tract. Okay? So the mucosa, we're going to find in the mouth, the pharynx, the esophagus, and the anus. These are non-keratinized, stratified squamous epithelium. So these are stacked up sort of pancake-like cells. And we always find that really to resist abrasion. In our stomach, our small intestines, and the large intestine, that mucosa is simple columnar epithelium. Okay? And that is for digestion and absorption. We also have goblet cells that are interspersed between those columnar epithelium, and they produce mucus for lubrication. Then we have the muscularis. Remember that muscularis is inner circular and outer longitudinal layers of smooth muscle, and that's where we can sort of milk that food out. And remember that action is called peristalsis. The serosa and the visceral peritoneum are the same thing, just two different names for the exact same structure. We have simple squamous epithelium, and it sits on an areolar connective tissue, which forms the basement membrane. Okay? And so we have this enteric nervous system, or our nerve supply. This mesenteric plexus It lies in this muscularis layer of the alimentary canal, and its job is motility, and that myenteric motility, both starting with the letter M, that can kind of help you with that, and it forces those contractions of the muscularis, also M, muscularis, right? So myenteric motility of the muscularis. And then we have the submucosal plexus. The submucosal plexus is in the submucosal layer, and it's responsible for regulating the digestive secretions. It reacts to the presence of food. Okay? So you can see that here. This is that myenteric plexus. We have an interneuron between to hit the submucosal, can hit those mucosal epithelium. And then we also have that myenteric plexus going straight here to the longitudinal and circular smooth muscle layers of the muscularis. Okay? So blood supply to our digestive system is going to come straight through that abdominal aorta. Okay? So we have our gastric arteries. This is all part of that celiac trunk, which you might remember from the cardiovascular system. The gastric artery, the splenic artery, and the common hepatic artery are going to branch off of that celiac trunk. Okay. And we continue to branch until we can hit each one of our organs that we need to. The superior mesenteric artery, what's key about that is that it's the first 2/3 of the large intestine. And then the inferior mesenteric is going to be the last third of that large intestine. Okay. This is the peritoneum. Okay. So what you can see here is this is the visceral peritoneum. Remember, the visceral part is going to be the part that actually wraps around the organ itself. Okay. And then we have the parietal peritoneum. That's going to be the part that covers the wall of the abdominal cavity. And between them, we have the peritoneal cavity. This peritoneal cavity is also called the peritoneum. Okay. And peritoneal fluid is serous fluid. That's really for lubricating to prevent friction, because remember, those intestines are going to undergo peristalsis. So the process of digestion is ingestion, propulsion, mechanical or physical digestion, chemical digestion, absorption, and defecation. And if you check out your textbook, you'll find tons of devices to help you. Something about an indigo parrot, but honestly, I don't remember what it is. So ingestion, propulsion, mechanical digestion, chemical digestion, absorption, and defecation. These are the processes of digestion. Okay? And so we can sort of see how we're actually doing all of these things throughout. So we don't just like do one and then move on to the next one. It's sort of a constant cycle. So obviously ingestion, that's putting the food in your mouth and ingesting it. But we actually start this whole chemical digestion right here in the mouth, as well as we start mechanical digestion directly in the mouth, right? So for mechanical digestion, that means it's the mechanical process. So if you imagine for a second a piece of paper, and I've just torn that piece of paper apart, right? So that's the mechanical part, is I'm just breaking it into smaller and smaller pieces. But what if I were to take that piece of paper and I were to put some water on it? Okay. And so that's going to become the chemical digestion. If you just put some water on some paper, it's going to start to break down even further. So chemical digestion is when we take those enzymes and they break down those chemical bonds. Okay. And so that, again, is starting right here in the mouth. Mechanical digestion and chemical digestion are happening here. Okay. We have propulsion, right? Propulsion is that active movement. Okay. The active movement down, right? So ingestion, propulsion, mechanical digestion, chemical digestion. We're talking about those right now, right? So propulsion is moving down, right? So the swallowing, for instance, at the oropharynx, the peristalsis, that sort of milking motion moving down, okay? And then we have absorption. And most of the absorption is happening in the small intestines. You can see it here. Okay? But we also have some water absorption happening at the large intestine. And then defecation. And defecation is when we remove that solid waste. So ingestion, it's occurring in the mouth. The teeth and the tongue are doing the mechanical digestion. The tongue is sort of moving around in your mouth to push the food back and forth from side to side. And then the teeth are sort of that tearing action, right? Then our salivary glands are the chemical digestion. The saliva is secreted from 3 of our salivary glands. We have the parotid gland right here up in the cheek. We have the submandibular gland at the bottom of the mandible, and then the sublingual, which is underneath the tongue. And then we have propulsion. So remember, propulsion is that peristalsis, but it's also segmentation. And really, propulsion has to do with the movement, okay? So once we swallow our food and we start to digest it, it becomes what we call a bolus, okay? So we no longer call it food. So this peristalsis happens, and it's a movement or a propulsion. Segmentation is another movement that occurs, but segmentation is a form of mechanical digestion. And this happens with a mixing, right? So we get sort of this segments that are forming, okay? And it's going to keep sort of moving back and forth, back and forth, back and forth. Okay? Then we have this mechanical digestion. So in the mouth, we chew the food, we make it into smaller particles. In our stomach, we actually have a grinding that happens, right? So we sort of grind we have 3 layers of muscular tissue along the stomach. And so we have a longitudinal layer and a circular layer. And those are going to be sort of squeezing in 3 different planes to really squeeze that food and make it smaller, make that bolus even smaller. And then our small intestines, we have that segmentation happening, right? So it's moving it back and forth, back and forth, back and forth, which is definitely a form of mechanical digestion. Okay. And then in the chemical aspects, we have enzymes, right? So enzymes, if you remember, they're going to have that ASE at the end. So we have amylase. Amylase helps to break down sugars, which are amylose. Amylose is a carbohydrate or a sugar. We have lingual lipase, ACE for lipase helps to break down lipids or fats. Then in our small intestines, we also have pancreatic lipase in there to help digest fats. We have amylase digesting carbs. And then we also have proteases, which are helping to digest our proteins. Okay. This is just another diagram. I think this one is directly from your textbook. So we have that propulsion, okay? So we have the swallowing and then the peristalsis action. We have our chemical digestion, which is our enzymes. We have mechanical digestion, so our chewing here, our churning, which is what our stomach does, and then our segmentation here. And then we have our absorption, so we're absorbing nutrients and water. So absorption is when we're actually pulling in all of the good nutrients from our food. Okay. So this is an image of the intestinal wall, and on the intestinal wall we have what are called villi. And those villi sort of look like projections coming out, right? So this would be like if you just imagine for a second that your fingers, right? So your fingers are sticking out, but then off of those fingers you also have what are called microvilli, and within your intestines, on your fingers, we call them fingerprints, right? And so those are there so that they can increase the surface area, okay? So the microvilli help us to increase that surface area so that we can digest more. Okay, so that happens on the apical surface of the mucosa in the small intestines. And our final digestive products, like for carbohydrates, are going to be monosaccharides. Remember that word, that prefix mono means one, saccharide means sugar. So it's a single sugar, really small. Proteins we can digest as dipeptides or absorb as dipeptides. Di means 2. Or amino acids. And fats, we're going to get fatty acid chains and monoglycerides. So then we have defecation. Hold on one second.
It's Sunday. Why are you doing work?
She wants to know why I'm doing work on Sunday. So defecation is when we're actually going to have that waste or the feces accumulated during the passage through the colon or the large intestine. Defecation happens through the anus, and we have 2 sphincters there, the internal and the external anal sphincters. So we have some neural controls, right? So we already talked about that plexus that's going to help us is sending those messages, okay? But we also have some nervous control of the GI tract, like when we have the sight or the smell or the taste or even the thought of food, right? So if you just think about bacon cooking for a minute, if you like to eat bacon, right? When you start to smell that bacon, you all of a sudden are going to get hungry, right? Somebody's woken you up with cooking breakfast in bed. How nice would that be? Never happened to me. But I can just imagine. So I would experience that smelling and then thinking about the food. And when that happens, we get a response, right? So we send a signal through an efferent impulse to our enteric nerve plexus. And then that's going to send a message out to our smooth muscle glands, okay? And they're going to start to change their contractile or their secretory activity, okay? We also get a stimulus here. So for instance, when we actually eat, okay, we get a stimulus because we're going to have those mechanical and chemical and osmotic stimuli are going to be here within the wall, okay? So those chemoreceptors, osmoreceptors, and mechanical receptors, they're going to send that signal back up. Okay? But this is a reflex response. So when we have our hormonal controls, the stomach is empty, right? Usually about 3 hours after a meal. Our gastric cells are going to secrete ghrelin. Gastric cells secrete ghrelin. And that's going to travel to our feeding cells within the hypothalamus, and it triggers our hunger center to stimulate us to take in food. So about every 3 hours, we get hungry. After the food has been digested and the adipose and the nutrients is depositing that fat, the adipose cells release leptin. Okay, and leptin is going to travel to the feeding cells in the hypothalamus. That triggers our satiety center, and it says, "Oh, we're full," right? So it's your fat cells that actually tell you that you're full. And then we have our peptide YY from our small intestines. They can also trigger satiety after we eat. Gastrin comes from our G cells within the stomach, okay, and those are going to secrete hydrochloric acid to help us with, to actually digest and do some chemical digestion. When the food enters the duodenum, we have these enteroendocrine cells, and they're going to secrete cholecystokinin. That's CCK. Okay? And CCK is going to trigger our gallbladder contraction so that our pancreatic enzyme release, so we can release our amylase to digest our carbs, the lipase to digest our fats, and our proteases to digest our proteins. And then secretin is going to trigger our pancreatic bicarbonate release so that we can neutralize gastric activity. Okay, so what happens is, remember, this is hydrochloric acid in here. Okay, but once we get into our small intestines, we don't want our bolus of food to still be covered over in acid. So that secretin helps us to release our bicarbonate so that we can neutralize that stomach acid. So after we eat, our blood glucose is going to rise above normal, and that's going to trigger those beta cells. Those beta cells are found in the islet of Langerhans in the pancreas, and those beta cells are going to release insulin, right? So after we've eaten, our blood sugar goes up, right? And so when our blood sugar goes up, our body releases insulin. The insulin is going to bind to our cellular receptors, and that triggers the opening of our glucose channels, so that those cells can pull in the glucose so that we can then use it for cellular respiration. When that happens, our blood glucose levels will return back to normal. When we fast or we're sleeping, so we're not eating or we're asleep, our blood glucose levels are going to fall below normal. And that's going to trigger our alpha cells, which are also found in the islets of Langerhans within our pancreas, to release glucagon. And glucagon is going to trigger the liver to produce glucose by gluconeogenesis and glycogenolysis. That's released into our bloodstream, and our blood glucose can then increase and go back into a normal range. Okay, so when we've eaten and we have a lot of sugar in our blood, we release insulin. When we're not eating, we have little sugar in our blood, we release glucagon. Okay. So this is just a chart to help to summarize out all of our major functions of our digestive organs. Here's the organs, here are the functions, and then some extra functions off to the right. Okay, so you should definitely become familiar with this chart. So within our mouth, we have different types of teeth. Okay? Specifically, we have some incisors here. The incisors are going to help us to bite the food. They use that masseter, which is right in the cheek, and our temporalis muscles. The molars, which are going to be back to the back, are going to grind up the food. They use the lateral pterygoid muscle. And that's the process of chewing. We also call chewing mastication. Mastication. Chewing increases the surface area of our food so that as we're doing mechanical digestion, we can then start to use our digestive enzymes, so do chemical digestion using amylase, which is in our saliva, okay, and our lingual lipase to digest those short fatty acid chains. Okay. So these are our teeth. Okay. So the yellow ones are our incisors. We have the way that you count your teeth is you actually find the center of your teeth, and then you're going to find the dental formula. Okay, so it's 1, 2, 3. Okay, so you have from the center, you have 2 incisors. So you have a central and a lateral. Then you have 1 cuspid or canine tooth. Then you have 2 premolars, first, second, and then you have 3 molars, first, second, third. We call the third molar also your wisdom teeth, so you may not actually have that. Okay, but we always start here from the center. These are the upper teeth, these are the lower teeth, but this is the same formula, 2, 1, 2, 3. Okay. This is the tongue. The tongue is made up of skeletal muscle, and those movements are required to position the food, also for talking, as I like to do. And it's covered with stratified squamous epithelium to resist abrasion from the food. At the base of the tongue, we're going to find the lingual tonsils. Okay? And the tongue surface is dotted with what we call papilla. Okay? And this is the fungiform up the center, and then we have the filiform papilla at the edges. Okay? We also have circumvallate papilla along the back. So the fungiform papilla are where we're going to find our taste buds. We call them crypts, which house the taste buds, and those have gustatory receptor cells. Okay, there's 5 types of gustatory receptors. Okay? We have sweet, sour, salty, bitter, and then the center we call umami, just savory. Okay? And so when we trigger that, the cells are going to transfer the information to our cranial nerves. So we have osmoreceptors, okay? And they're going to sense that sort of change in the chemicals, and we're going to send in the signal. So the facial nerve is going to be for the anterior 2/3 of the tongue, and then the glossopharyngeal is going to act on the posterior 1/3 of the tongue. And then in the back of your throat, we have the vagus nerve, which is going to be our posterior pharynx. Okay, so these are all of your cranial nerves. Hopefully you remember those from Anatomy 1. And then that information is going to be analyzed within our insula lobe of the brain. Okay. There's 3 types of salivary glands. There's parotids. Parotids are located again in the cheeks, the submandibular under the mandible, and then the sublingual under the tongue. Saliva is mostly water, but it also has mucin in it, and that mucin is for lubrication of the food. We have bicarbonate. Now, bicarbonate is going to maintain a neutral to basic pH, about 6 to 8, which is going to help to neutralize our stomach acid that might enter the mouth or the esophagus. We have salivary amylase in here. Salivary amylase is going to help to digest carbohydrates. Lingual lipase is going to digest the fat. We also have lysozymes and IgA. Remember, these are the immunoglobulin A antibody, and that's present for our immune response. And the peripheral nervous system via our glossopharyngeal and vagus nerves are going to trigger salivation. The pharynx is going to run from the nostrils to the esophagus and the larynx. And remember, in the back is the esophagus, and the front is the trachea. So the esophagus is that tube. It's going to travel all the way down the thoracic cavity, and it goes behind the trachea. It starts at what we call the upper esophageal sphincter, and it ends at the lower esophageal sphincter just before going into the stomach. The mucosal lining is stratified squamous epithelium, and that helps to prevent abrasion. We have a muscularis layer up here for the upper third. That's skeletal muscle, and then smooth muscle for the lower 2/3. Because remember, you have control here. That swallow. So this swallowing mechanism, we call it as deglutition. Deglutition. So the first part is voluntary, right? So that bolus is being swallowed. We're going to have the uvula moving up to cover over that nasal cavity. We're going to find that the epiglottis is going to fold down to cover over the trachea. and that's going to allow that food to move down. Okay? Then in the pharyngeal stage, we're in the pharynx. So we start to constrict the muscles. You can see them here. And then the esophageal is when we have these constrictor muscles, they're going to start to constrict going down. This is peristalsis. We've already covered that, but just a quick review is that we have this sort of wave of contraction. We also can see segmentation, which is a mechanical digestion. Okay? So to review, let's discuss the processes and the control of ingestion, propulsion, mechanical digestion, chemical digestion, absorption, and defecation. So pause here and give it a try on your own. So these are those 6 processes of digestion. Okay, we have ingestion, mechanical digestion, chemical digestion, secretion, absorption, defecation. So secretion, not really that process of digestion that we talked about, right? Ingestion, mechanical digestion, chemical digestion, absorption, defecation, propulsion. So this is our stomach, okay? So here you can see those 3 layers of muscular tissue that I talked about. You have your longitudinal, your circular, and your oblique layer, okay? This is the cardia right here. This is where our esophagus enters. Then we have the fundus. The fundus is the dome-shaped area that's going to fit right under the diaphragm. The body is going to be the main portion. The antrum is in here. This is an area of propulsive force, and then we have our pylorus, which is the sphincter where the food is going to exit. So the food's coming in here, and then it's going to go right there. When our stomach is empty, the mucosa is going to fold up into a pattern which we call rugae. Okay. And so what's cool about our stomach is that because we have these 3 muscular layers, it's going to churn the food. Okay, so it contracts in 3 different planes. And as it does that, it's going to force to turn the food into a paste. And that paste, remember, we call it chyme. So within our stomach, we have what are called gastric pits. Okay, here's a gastric pit right here. Below the pits are going to be the gastric glands. Okay. And within them, we have the enteroendocrine cells. Those are the ones that are like the G cells in the antrum. They help to release the gastric juices. Okay, so these are the parietal cells and the chief cells. Okay? The parietal cells are going to secrete hydrochloric acid in response to gastrin. The chief cells are going to produce pepsinogen, which when we have it in the presence of hydrochloric acid, which you do in your stomach, it's going to activate into a protease called pepsin, and pepsin is responsible for about 10% of our protein's digestion. So take a second and see if you can fill in this chart. So the hydrochloric acid, with its pH of 1, it's going to play lots of different roles within the stomach. So first off, it's going to help to kill pathogens within the food. Okay? It also helps to denature proteins. So if you remember with proteins are amino acid chains that have bound and wound on top of one another. I always give that visual of a curling ribbon that you might use to wrap a present. So you can stretch it straight out, or you can pull the scissors across it. And when you pull the scissors across, it's going to start to form bonds and start to curl over itself. You can add more than one color to your ribbon. Okay, and so that would be the 3D structure of a protein, right? It's all curled over on top of one another. Okay, but what hydrochloric acid does is that it breaks those bonds. Okay, it breaks the hydrogen bonds that are going to create that 3D structure, and that unwinds the ribbon, or it unwinds these proteins, and that helps to chemically digest the food. It's also going to activate that pepsinogen into pepsin, and it's required to remove our vitamin B12 from and then attach it to intrinsic factor, which can help it to facilitate absorption. So we needed to absorb the vitamin B12. So there's 3 different phases of our gastric secretion. So the cephalic phase, cephalic has to do with your brain, right? And so this is when we have the sight or the thought of food. And then we can start to stimulate the taste and the smell receptors. Okay, that's going to stimulate our stomach secretory activity. Okay, it's also going to inhibit sort of when we have a loss of appetite, like with depression, our cerebral cortex is going to have a lack of stimulation to our parasympathetic center. Okay, the gastric phase here, okay, is when we have a stomach distension. Okay, that's going to stimulate our stomach secretory activity. And then we have our intestinal phase. And during our intestinal phase, we have a presence of a low pH, right? Because it's going to have been covered over in our stomach acid. It's going to move into our duodenum as our stomach empties. And that's going to stimulate, that shouldn't say stomach. It's going to stimulate our gastrin release into our blood. So gastric motility, remember we have that peristalsis, okay? We have our oblique, circular, and longitudinal layers. They're going to create the mixing waves, and they occur about every 20 seconds. We have these waves that are going to crescendo within the antrum, and that has the most propulsive force, that it crashes and crushes the food into that chyme. During our gastric emptying, only about 1 teaspoon of chyme exits at a time, so it takes about 3 to 4 hours for the stomach to fully empty. The small intestine can only handle small amounts at a time. Any more, and diarrhea would result. And fatty foods are going to delay the gastric emptying. So if you eat something with a little bit of fat in it, it keeps you fuller longer. Okay. This is our small intestine. The small intestine is about 10 feet long within adults. Duodenum, the jejunum, and the ileum. Okay. The duodenum is about 10 inches long. The jejunum is about 3 feet, and then the ileum is about 6 feet. Okay. The blood supply, remember we have that superior mesenteric artery. We have the vagus nerve, and we also have MALT. Remember that mucosa-associated lymphoid tissue for our immune response. This is the duodenum. The duodenum is C-shaped curve, and it curves around the head of the pancreas. It's where we have those enteric endocrine cells that are going to secrete the CCK to trigger our gallbladder to release its enzymes and the pancreatic enzyme release, and then secretin is going to trigger our pancreatic bicarbonate release. We also have what's called the ampulla here, okay? And this is the, it's the ampulla vator is going to enter the pancreatic secretions, like the amylase, lipase, proteases, and bicarbonate, and the bile. And it's where we have the Brunner's glands, and the Brunner's glands are going to secrete alkaline fluid to neutralize our acidic chyme. Then we have the jejunum. The jejunum is our primary site of digestion and absorption. And within here, we have what are called crypts of lubricant. They're going to secrete 1 to 2 quarts of neutral pH fluid daily so that our chyme is not hypertonic. And then the ileum, which is our lowest level of our small intestine, is going to finish our digestion and absorption. It's the site of our bile acid reabsorption for recycling, and then also the site of vitamin B12 absorption. It ends at what we call the ileocecal valve, okay, which would be right here. Within our small intestine, remember we have the villi and then the microvilli. Okay, we call that microvilli also a brush border. You may hear it referred to as a brush border. Okay, and this is where we actually digest our simple sugars. Okay, so disaccharidases, which are enzymes to digest disaccharides, and peptidases to help to digest di and tripeptides. Remember the colon is to reabsorb our 1 to 2 quarts of water daily. Okay? It's the home of 3 trillion bacteria, which is where we have our normal flora. Those normal flora helps to produce vitamin K. Vitamin K is needed to convert prothrombin to thrombin in our blood clotting. It helps to digest our functional or soluble fiber into fatty acids. And it helps those, they also unfortunately produce gas by doing that, although most of that gas that you may have or flatulence is swallowed air, and they help to crowd out invasive pathogens, right? So they are in a contest with any invasive pathogens. So if they take up the resources, they're already there, they've already made home, then those invasive pathogens can't make a home within your intestines. And it's also the site of those densely packed Peyer's patches. Remember we talked about Peyer's patches in our immune system. Okay? They help to prevent a bacteria from moving into the bloodstream. And then we have defecation. There's different sections of our colon or our large intestines. We have our cecum, the colon, the rectum, and the anus. Okay, so the cecum is the first part here. There's the appendix right there. So the cecum is basically just a holding zone. Okay, it's also where we find the vestigial vermiform appendix. Then we have the colon. So there's the ascending colon, the hepatic flexure, the transverse colon, the splenic flexure, the descending colon, and then the sigmoid colon. Okay? And the sigmoid colon is going to produce that propulsive force for defecation. And then the rectum. The rectum is just a holding zone for the feces until we can defecate. The rective valves, which you have an internal anal sphincter and an external anal sphincter, they help to separate the feces from the gas. Okay. And so look here is that we have a dietary input of about 2,000 milliliters. We then secrete about 1,500 milliliters. And then we have a gastric secretion of about 1,500 milliliters. When you add those numbers together, you end up with about 5,000 milliliters of fluid coming into the stomach. Okay. The liver is then going to secrete about 1,000 milliliters. The pancreas juices secrete another 1,000 milliliters. Our intestinal secretions another 2,000 milliliters. So here in the intestines, we're looking at about 9,000 milliliters of fluid. But the small intestine is going to reabsorb about 7,800 milliliters. So if you take 9,000 minus 7,800 milliliters, we're left with about 1,200 milliliters that is going to enter into the colon. The colon is going to secrete about 200 milliliters of mucus. But then look at this. The colon reabsorbs 1,250 milliliters. And so only 150 milliliters is going to be lost within the feces, right? Because it's really important that we reabsorb that water. We don't want to lose the water. On our colon, we have what are called teneocoli. There they are. And these are 3 bands of smooth muscle along our longitudinal muscle layer of the muscularis. And that gives some tonic contractions of that teneocoli. It's going to bunch up the colon into these pouches, which we call haustra. And that gives us the wrinkled appearance of the colon. We also have epiploic appendages. Okay. Those are adipose globules in the visceral peritoneum, and those are attached to the teneocola, but we have no idea why. This is looking at the histology under the microscope, right? And so you can see all of these goblet cells in there. Those goblet cells are all the little white dots. Okay? They secrete that thick mucus to lubricate our solid stool. And there's no villi here because we are not digesting anything. We're just absorbing water and vitamin K. So we have stratified squamous epithelium. Remember, I told you every time we time that, stratified squamous epithelium is to resist abrasion, okay? And it's to resist abrasion from our solid stool during defecation, and it merges to that of our outside skin. We have a superficial venous plexus. That superficial venous plexus, unfortunately, sometimes can get inflamed, and that will give us hemorrhoids when we have any extra straining or excessive weight bearing. And the anus is going to contain the internal and external sphincters that are involved in defecation. For defecation, the feces is going to fill the rectum, any undigested fiber, millions of bacteria, old epithelial cells, inorganic salts, and enough water, about 150 milliliters, to let it pass smoothly out of the body. Little, if any, nutrients are going to remain in this because we wanted to absorb all of those nutrients. Okay, we've already talked about the Valsalva maneuver. We talked about the respiratory system. But we can also use that Valsalva maneuver to enhance defecation because it increases our intra-abdominal pressure. We contract the diaphragm and the abdominal wall muscles while closing off the glottis. Okay, that's when we're sort of straining. And our false cords are going to flap over the true vocal cords, so that prevents any air from escaping. The feces are going to accumulate in the rectum, and that results in the feeling of a stretch, okay? And so the internal anal sphincter is going to sense that stretch. It's going to relax to initiate defecation. This is under control of the parasympathetic nervous system, right? Rest and digest. In the sigmoid colon, it's going to use the motility so that we can move the feces along. And then the external sphincter is under our voluntary control. You can override the internal anal sphincter when necessary, but only for so long. When you have to go, you have to go. These are those accessory organs that I talked about, right? So we have the liver, we have the gallbladder, we have this the spleen, the pancreas, right here. So the liver is located up in the right upper quadrant of your abdomen. It has a large right lobe and a small left, and it's easily accessible for removal for transplant. We have the porta hepatis, and this is where the hepatic artery, the hepatic portal vein, and the common bile duct are going to enter into the liver. Okay. On the cellular level, we have it arranged in lobules. Okay. And so you can see what happens is this is from the portal vein right there. You can see all those lobules. This is a lobule here. And so we have our interlobar vein. Okay. This is a central vein going right down the center. Okay. And so we kind of bring the blood flow up. Okay. And so the fenestrated capillaries with the blood from the nutrient-rich hepatic portal veins and the oxygen-rich hepatic arteries are how we're going to get the absorption of the nutrients here in the liver. So our blood from our portal vein is going to enter into the portal venules on the edge. The blood is going to pass through the sinusoidal capillaries, okay, where the liver cells are going to process the nutrients, and then it's going to distribute to the body via our central vein, and that's going to go from to the hepatic vein, which is then going to enter into the inferior vena cava. We have what's called a portal triad. That portal triad is the portal venule, the portal arteriole, and the bile duct. So the liver's job is to process the nutrients, and then it's going to distribute them out to the body. It also produces the bile to be stored in the gallbladder. It can metabolize toxins. It has Kupffer cells. Kupffer cells are going to remove any bacteria arriving from the intestines. Any idea what else they might do? They help to break down old red blood cells. They store glucose as glycogen. What's that process called when we store glucose as glycogen? Anybody? Glycogenesis. We're going to use ferritin to store iron from our degraded red blood cells, and we help to degrade hemoglobin into bilirubin for export in the bile. Okay? So let's talk about bile. Bile is composed of bilirubin, cholesterol, and bile acids. So bilirubin is the green pigment, okay? And it is a leftover from our pulling apart those old red blood cells. We have a product of hemoglobin, okay? It is formed within the spleen and the liver. The liver excretes it into the bile. And in the intestines, we can turn it into stercobilin, and stercobilin is the brown pigment of stool or poop. Okay. The cholesterol is removed from our dying cells by what are called HDL lipoproteins. It's excreted in the bile, and it lowers the risk of atherosclerosis. And then bile. Bile acids are phospholipids. Remember, phospholipids are glycerol with an attached phosphate group. I have to peel an orange. And remember that oil and water don't mix. So within our intestine, we have these hydrophobic tails. They're going to surround the triglycerides while the hydrophilic head dissolves within the intestinal juice. This allows the lipase to access the triglycerides and digest it into a monoglyceride, and the two fatty acids are then going to be absorbed, right? So they help to sort of break apart fat globules, okay? We call it bile acids or bile salts sometimes, right? And so if you just imagine this really large piece of fat, it can sort of start to break it and split it apart. And when we can split it apart, it just makes it easier to digest, okay? There's different shades of poop. So brown is normal. That's considered fine. Okay. Green is when our food might be moving through our large intestine too quickly, or you could have just eaten a lot of green leafy vegetables or even green food coloring. And then the yellow is usually greasy, foul-smelling yellow poop. It indicates that you've had excess fat, and that could be due to a malabsorption disorder like celiac disease. Okay. The gallbladder, it's tucked up underneath the right lobe of the liver. The bile from the right and the left bile ducts are going to merge into the common bile duct. Okay, so here's the gallbladder. This is the common bile duct. Okay, so you have your common hepatic duct there. Okay. You have the left going to the left side of the liver, right going to the right side, and this is the cystic duct. Okay, so what happens is we're going to lead it here into the cystic duct to go into the gallbladder. The gallbladder concentrates the bile. Okay, CCK is going to trigger the action of the gallbladder to release the bile into the common bile duct. Okay. Then the bile is going to enter the duodenum through the ampulla of Vater. Okay. So there you can see that common bile duct here. Okay. So the pancreas is retroperitoneal, meaning that, remember that peritoneum is going to separate the pancreas secretions from ever being released within the abdomen. The pancreatic duct also is going to empty through the ampulla of Vater into the duodenum. Okay. So remember, our pancreas has those islets of Langerhans. We have the alpha cells that are producing the glucagon. We have that when our blood glucose is low. Okay. Glucagon triggers the liver to perform gluconeogenesis and glycogenolysis, right? So if you think about it, when you're fasting and you don't have enough sugar in your blood, we can do gluconeogenesis. That means that we're going to take our fatty acids, we're going to take our amino acids, we're going to bind them together so that we can form glucose. Okay? And then we can also do glycogenolysis. Remember, glycogenolysis is when we break apart glycogen into the glucose. And then we have our beta cells producing insulin. Okay. Beta cells are going to produce the insulin when our blood glucose goes high. So just after a meal, right? When we've eaten and our blood glucose goes up, okay, we need to get that blood glucose or the glucose that's in our blood into the cells so that they can do cellular respiration. The acinar cells within our pancreas, this is our exocrine pancreas, they're going to produce our digestive enzymes and our bicarbonate. The digestive enzymes is going to produce amylase. Amylase is going to digest our complex carbohydrates into glucose and maltose. Lipase is going to digest triglycerides into monoglycerides and 2 fatty acids. And proteases, we have bunches of different ones. Okay, we have trypsin, they're going to digest a bunch of them. Okay, but what we have is proenzymes, and proenzymes are going to be formed, and they must be activated. Okay, the brush border enzymes are going to activate them, right? Because if we just release proteases, they would just start to digest proteins, and most of your body is made up of proteins. Proteins, remember, enzymes, those are the things, they're proteins. So if we just released a whole bunch of proteases, enzymes that are going to break down proteins, then it would break down the body itself. Okay, so we require them to be activated. Okay, so we release proenzymes, and those proenzymes are going to have to be activated. They get activated at the brush border. And that brush border, remember, is in that small intestine along the microvilli. This exocrine pancreas is going to release bicarbonate. Okay, that's a pH 7 to 8, and that neutralizes our acidic chyme in our duodenum, and that's needed to activate those proteases as well. Okay, so take a minute and do this quick review. See if you can do it on your own. So when we list and describe the functional anatomy of the organs and the accessory organs of the digestive system, right? We have the salivary glands, the pharynx, the esophagus, the stomach, the small intestine, large intestine, the rectum, and then these are accessory organs. So give it a try and see if you can describe what each thing is doing and what about each part does its job, like what's functional about the anatomy. Okay? The roles of the liver, the pancreas, and the gallbladder in digestion. Here we go. And then compare and contrast the digestion of the 3 macronutrients. Okay? So these are carbohydrates, proteins, and fats. So just give it some consideration as to what's each part doing, right? Where is it happening and what is the absorption, okay? So when we look at carbohydrates in the green, we started as starch, disaccharides, right? We just start to break them down further and further into the monosaccharides, okay? Like glucose, fructose, and galactose. Okay? Then we have our protein digestion, right? So we start to break it down within our stomach. We break down pepsin in the presence of our hydrochloric acid. It's going to happen. We have our pancreatic enzymes within the small intestine. And then in order to get to the smallest part, our amino acids, we do that by activating all of these different proenzymes to break down even further. And then we have our fats. So we have unemulsified fats. They're going to be emulsified. Emulsified is that term of the breaking them apart with the salts, the bile salts. Okay. And we're going to break them down into monoglycerides and fatty acids, and then the glycerol and the fatty acids. Okay. All right. So if you have any questions, you guys know where to find me. Send me a quick email, and I would be happy to help. Bye.